Dear all, how are you? Today, I will talk heavy metals pollution in Chapter 11.
Unlike other pollutants, which may visibly build up in the environment, trace metals in the environment may accumulate unnoticed to toxic levels. Heavy metals are commonly defined as those with a specific density of more than 5 grams per centimeter cube. Heavy metals are serious pollutants in our natural environment due to their toxicity, persistence, and bioaccumulation problems. Heavy metals, in aquatic systems, can be from natural or anthropogenic sources. Currently, the anthropogenic inputs of metals exceed natural inputs. Organisms require trace amounts of some heavy metals, including cobalt, copper, iron, manganese, and zinc. However, the excess metal levels in aquatic may pose a health risk to humans and the environment. Excessive levels of essential metals, however, can be detrimental to the organism. Non-essential heavy metals, of particular concern to surface water systems, are cadmium, chromium, mercury, lead, and arsenic. Water chemistry of the system controls the rate of adsorption and desorption of metals to and from sediment. Adsorption is a process of removing the metal from the water column and stores the metal in the substrate. Meanwhile, desorption is a process of returning the metal to the water column, where recirculation and bioassimilation may take place. Metals pollution has drawn increasing attention worldwide due to a dramatic increase of anthropogenic contaminants to the ecosystems through air, water, and soils. Pollution of the natural environment by metals is a serious problem because these chemical elements are indestructible, and most of them have toxic effects on living organisms when they exceed a certain concentration. Some metals are highly toxic and persistent, and have a strong tendency to become concentrated in marine food webs. The accumulation of heavy metals in superficial sediments, from industrial effluents, and urban sewage discharged into the aquatic environment, without proper cleaning, will easily be identified through metals' spatial variations in sediments. Geochemical mapping can be used as a tool for visualization, which enhanced by computer-aided modeling using Geographic Information System (GIS) to make it easier to identify the possible locations of contaminated area. This is one example how GIS software can be used to mapping the hotspot of polluted area. Heavy metal concentrations in harbor or estuarine sediments usually are quite high, due to significant anthropogenic contaminants loading carried by the upstream of tributary rivers. Sediments can serve as a metal pool that can release metals to the overlying water via natural or anthropogenic processes, causing potential adverse health effects to the ecosystems. Lying in the second trophic level in the aquatic ecosystem, shellfish species have long been known to accumulate both essential and non-essential metals. Many researchers around the world have reported the potentiality of using mollusks, especially mussel and oyster species, as bioindicators or biomarkers for monitoring the metals contamination of the aquatic system. Besides serve as a biomarker for marine pollution studies, mollusk species have also been used in ecotoxicology and toxicity studies. These figures show some of the shellfish that used as bioindicator or biomarkers for pollution studies. When we study the pollutant in the organism, the safety of food to the consumer is a major concern, to ensure the safety of consumers. Therefore, metals analyses of the aquatic organisms can provide important information on the degree of environmental contamination and potential impact of seafood, especially fish and shellfish consumption. Heavy metals analysis. The concentration of contaminants in sediment and organisms are expressed as microgram per gram or ppm, and microgram per kilogram, ppb. For sediment, nominally based on dry weight. For organism, either can be wet weight or dry weight of tissue. What are the sources of heavy metals in our environment? Heavy metals of natural and/or anthropogenic origin is transported by rivers and transferred to the coastal marine system through the estuaries. These pollutants can enter the coastal environments and oceans by two means. First one is natural processes. For example, erosion of or bearing rocks, wind-blown dust, volcanic activity, and forest fires. The second one is from human activities, such as atmospheric deposition, rivers, and direct discharges or dumping, wastewater. Marine aquatic organisms can accumulate heavy metals from various sources in their surrounding environment. Once the heavy metals enter the food chain, they may accumulate to dangerous levels and be harmful to human health when consumed the polluted organisms. Heavy metals accumulation depend on several factors, such as environmental concentrations of metals in water and sediments, species of organisms, body size, and age of the marine organisms. Different concentrations of metals can also be found in different organs such as stomach, gill, muscle, liver, in the same biological sample. For example, in fish sample. Beside that, the entry may be a result of direct discharges into both freshwater and marine ecosystems, or through indirect routes, such as dry and wet deposition and land runoff. Important natural sources are volcanic activity, continental weathering, and forest fires. The major part of the metals compound in the water and the seabed sediments is anthropogenic and associated with terrestrial sources related to untreated municipal sewage, industrial wastewaters, intensive aquaculture. Harbor activities, river influxes loaded with urban and agricultural runoff, crude oil exploration and exploitation, emissions from vehicular traffic gas exhausts. Rivers flowing through urban areas may bring the pollutants to the downstream estuarine sediments, from where they are incorporated in the mudbank cycle. This process may result in downstream such as estuary ecosystem and higher concentrations of pollutants. How can marine sediment act as a marker for pollution studies? Sediment are widely used as geomarkers for monitoring and identifying the possible sources of pollution in the coastal environments, since sediments are the main sink for various pollutants. Most metals are bound in the fine-grained fraction, or less than 63 micron meter, mostly because of its high surface area to grain size ratio and humic substance content, where they have a potentially greater biological availability than those in the larger sediment fraction, from size of 63 micrometer up to 2 mm. Sediment cause can provide chronologies of contaminant concentrations and a record of the changes in concentration of chemical indicators in the environment. The metal accumulation rates in sediment cause can reflect variations in metal inputs in a given system over long periods of time. Hence, the study of sediments core provides a historical record of various influences on the aquatic system by indicating both natural background levels and the man-induced accumulation of metals over an extended period of time. Next, I will explain about biomonitor of heavy metals. Individual biomonitors respond differently to different sources of chemical elements, for example, in solution, in sediments, or in foods that they eaten. Therefore, we have to use a correct biomonitor that can reflect the element bioavailability in all available sources. Ideally, species to be chosen as biomonitors should fulfill several criteria, such as should be sedentary, easy to identify, abundant, long-lived, available for sampling throughout the year, large enough to provide sufficient tissue, resistant to handling stress caused by laboratory studies of metal kinetics and/or field transplantations, tolerant of exposure to environmental variations in physico-chemical parameters such as salinity. Living organisms can transport pollutants and contaminants into, within, and out of the marine aquatic ecosystem. These organisms can ingest the pollutants via water and food and inhale them as they breathe in feeding. Once pollutants in the body, some contaminants pass quickly, while others can be retained for long periods and accumulate in body tissues. The choice of a suitable biomonitor needs to consider the potential sources of metals to the biota. For example, seagrass, not in contact with sediments, will take up heavy metals from dissolved sources only. Suspension feeders such as shellfish organisms take up metals both directly from seawater and from the suspended particles collected during feeding. These are some of the organisms that commonly used as researchers as a monitoring agent. How can we access the pollution status? To evaluate the heavy metals contamination, determined element concentrations were directly compared to the background concentrations. Literature data on average world shale, or sediment cause, or sediments from pristine, non-industrialized regions were analyzed to establish the pollution status. The other approach is calculate the enrichment factors. Enrichment factors, or EF, can be calculated from heavy metals divided with reference in our sample, over heavy metals divided with reference in the background. Normally scientists use elements such as aluminium, lithium, cobalt, iron, scandium, cesium, and thorium as the reference in the calculation, and using the world average shale as the background data. After calculate, we can determine the contamination category based on the F value. If the EF value less than 2, we categorize it as deficiency to minimal enrichment. However, if the value more than 40, it fall under extremely high enrichment. The second approach is the calculation of index of geoaccumulation, or IGO. IGO can be calculated from log 2 of Cn, the concentration of the examined element, and divide with 1.5 times Bn, geochemical background concentration of the element, N. Why we need to times with 1.5? Because 1.5 is background matrix correction factor due to the lithogenic effects. In IGO, scientists group the sediment quality to 7 classes, from class 0 to class 6. In class 0, the IGO value were less than 0 or negative value and categorized as practically uncontaminated. However, in class 6, the IGO value more than 5 and can be categorized as extremely contaminated. The other approach is the calculation of pollution load index, or PLI. PLI can be calculated from square root of CF, depend on how many metals that we study. And the CF can be defined as heavy metals concentration in our sample divide with heavy metals concentration in background. The same average shale background can be used, similar to what is mentioned in the EF calculation. Several sediment quality based on the PLI value. If the PLI value less than 1, we categorize the sediment as low contamination factor. However, if the PLI is more than 1, it is considered polluted. So, what is the effects metals on human health? This figure shows how the contaminants in sediment move up to the food chain. The contaminant that enters the aquatic environment will settle down to the sediment. Then the shellfish organisms will uptake the contaminants through sediments and particulate. Fish will eat the shellfish, so the contaminant will transfer to the fish. The predator species, bigger fish, will uptake the contaminant from the prey. Lastly, the contaminant will uptake by human when we consume the fish. Generally, human health problems associated with trace metal contamination have been well highlighted in the literature. In spite of the relatively low level of industrial activity in less developed regions, there is nevertheless a high potential of toxic heavy metal pollution. Bioaccumulation, another way in which pollution can harm the environment, is the internal accumulation of a pollutant that is very difficult to degrade or expel from the body. Every organism accumulates pollutants during its lifetime, and if it is eaten, the pollutant are transferred to the predator or to higher trophic. Hence, bioaccumulation especially targets the top predators of the food chain, including humans. Do you notice that? Humans are the top predator in all environment? In marine, in land, in air. This figure also shows the flow how the contaminants uptake by organisms through primary organisms. In this case, algae is the primary organisms, and they uptake the contaminants through water column. And lastly, pasta human if we consume the organisms from the ocean. Let's look for one example. Oysters can be swallowed or masticated normally, increasing the surface of contact between food and digestive fluids. The consumer will consume the whole soft part of the oyster. Therefore, metals content is examined in toto or oyster flesh. One of the most dangerous metals that occur in our environment. Mercury. Mercury is very low level in the environment, less than 0.1 ppm. The permissible level in seafood normally not more than 0.4 ppm. The concern of mercury pollution starts from the incident of the Minamata Bay, Japan. The disease was first detected in year 1956, but the mercury emissions continued until year 1968. In Minamata, Japan, inorganic mercury was used in the industrial production of acetaldehyde, and discharged into the nearby bay, as wastewater, and was ingested by organisms in the bay. Fish and other organisms in the sea were contaminated, and eventually, residents consumed the fish suffered from methylmercury intoxication, later known as the Minamata disease. But even after the emission of mercury stopped, the bottom sediment of the polluted water contained high levels of this mercury. So till now, the impact is still visible.
That's all for this chapter. Thank you. Have a nice day.